Well, as always, it's a great pleasure to welcome you for this webinar organized by the Global Virus Network, and great pleasure to welcome Professor Damian Purcell. Just a few words, as usual, regarding the Global Virus Network, because some of the attendees do not necessarily know it so well. Just to remind you that the Global Virus Network is really a science-driven and independent network comprising now 57 research centers worldwide and 11 affiliates. And it's really a network which intends to merge the best experts in virology in a nimble organization, which is very complementary to the other national and international institutions, focusing on science, focusing on expertise, which is a key point nowadays, focusing on education and training with the project of the GVN Academy, very much organized around partnership with industrial partners, and very much involved in the advocacy. And on the website of the GVN, you can find a number of statements also and updates on different topics. So obviously, COVID-19 is at the forefront of our activities. And I might emphasize the biobanking effort that we are conducting and other efforts. And also the fact that 2 weeks ago, we had the annual workshop of the Global Virus Network fully dedicated to COVID-19 and an update on these pandemics. So in this context, webinars, and I thank Lynn Manley for this, have been really an important tool to, I would say, promote these collaborations, these interactions, and also the dissemination, I would say, of knowledge. And in this context, it's a great pleasure to welcome Damian Purcell. Damian is professor in virology at the University of Melbourne and the Peter Doherty Institute. He's in fact the theme leader for viral infectious disease at the Peter Doherty Institute and the head of the molecular virology laboratory. Damian has been trained in Melbourne, where he obtained his PhD, went to the NIH in the laboratory of Malcolm Martins for his postdoc, then came back to Australia, initiated his work in Australia in the Burnet Institute, then moved to Melbourne University and to the Peter Doherty Institute. Damian is a highly recognized international expert on HIV, HTLV1, deciphering the molecular virology and the mechanisms of replication of these viruses. But in addition to this, he is very committed to I would say, collaborations and to interactions. He's leading a number of initiatives in Australia, and he has been the past president of the Australian Society for Virology. And finally, Damian is highly committed to the Global Virus Network, and we very much appreciate his, I would say, positive interactions and strong support. So it's really a great pleasure to welcome you, Damian, and to listen to your lecture.
Thank you so much, Christian. It's a great pleasure to present to the GVN and anyone else attending tonight or early this morning, wherever you are. So I'm, as Christian was saying, from the Peter Doherty Institute, and we're very privileged to host the GVN meeting back in 2017, I think it was now. So this wonderful building is where we conduct our research, but we also have a function for education, so teaching of medical students and science research students, and importantly, public health function. So all of these things come together in a way that really assists in providing an opportunity when something like the pandemic comes along. And what I'm going to be talking about tonight is how the public health genomics effort that was really set up to understand the transmission of virus through our community in Australia, and particularly in the state of Victoria, where we're based, But this has provided quite unique opportunities for us to monitor the viral infection and to also build the consciousness of what's happening in terms of the evolution of the virus into our work, particularly focused towards developing effective vaccines. So Australia's had a fairly easy ride with COVID-19, not because of too many things other than very stringent public health measures, social distancing, etc. So we've had 2 outbreaks. The first outbreak, first wave, early in the course of the year, as familiar with many other people. But then through our winter in particular, instead of the distribution of virus outbreaks occurring mostly across all states in Australia, it was pretty largely confined to the state of Victoria. And so we at the Doherty Institute were in the epicenter of this second wave that really has been quite dramatic, and we're still actually in the tail end. We are in still severe lockdown, if you like. We're got a curfew at night with a 5-kilometre radius limit for our travel movements and limits on attending work, etc., social distancing. So very severe measures have really curtailed this second wave. But it's led to 27,206 cases with nearly 900 deaths, sadly. So the second wave, mostly in Melbourne, and a result, as I'll tell you in a moment, about resulting from mostly breach in return travel quarantining. Case numbers at the moment are less than 20 cases per day, and we're trying to get them down to fewer than 5 cases per day to break away from this lockdown. So we've got very high rates of testing, a low positivity rate, still about 0.6% in Victoria currently. And this is rapid, and we get an early diagnosis. You get your result pretty much the next day, if not the same day. And public health has really been prioritised as the significant response. So because of this, we've had a very good outcome. Australia here in this purple line, we've managed to curtail towards the end of our winter period as we come into our spring and summer, where hopefully we're able to enjoy a more normal summer season. So the response has been a very significant follow-up from the PCR testing done for diagnostics to follow up with genomics. So a very large amount of tests are done in the Victorian Infectious Disease Reference Laboratory at the Peter Doherty Institute. And this has sort of led into a genomics initiative, which has provided a very deep level of understanding of transmission virus. So infection, as you can see here, and there's a sort of publication in Nature Communications from Torsten Seaman and collaborators, Ben Howden, Narelle Cherry and others. The initial cases were almost exclusively return travellers coming overseas, so many strains, so a lot of differences that were very effective at enabling us to follow the particular transmission chain. So this was a very effective public health measure. So the work for us in terms of mutations with the ubiquitous presence, I guess, that occurred initially in Europe and then arriving in travellers from Europe was the D614G mutation. So, this is showing a period through May and June. So most of this first wave was within this group of return travellers, with the 614G mutation making a significant appearance within Australia. So this is sort of quite well known now, the impact of this particular mutation impacting on the access of the receptor binding domain. But we thought that this was, this is quite a dominant mutant within the spike region. many other common mutations arising in other parts of the viral genome and evident across different parts of the genome, full genome sequences, but really, quite lower levels of other mutations dispersed through that spike sequence. But later, as the outbreak sort of continued on into this period of the second wave, I hope what you can see here is that there's been a very significant appearance of spike mutant viruses. So spike mutant viruses that really popped up quite prolifically. So I'll come back to that previous slide, but if we look at these, so we've started following to see what these mutations were. So across the whole spike sequence, the particular region which is mostly focused, the main focus, if you like, of these of the concern, particularly for interactions with naive cells, so particular, alternative tropism, if you like, the ability to infect out of bat, pangolin, and then into human, is thought to be distilled in the receptor binding domain and the receptor binding motif. So we're quite interested in mutations here. The 614G is outside of this region, and as we looked across the sequences coming through the genomics program at the Doherty, and we're talking about more than 15,000 sequences, so 8,000 across Australia, 7,000 in the Doherty. We see that there are many individual single point changes that occur right across here. So there's a constant sort of trickle of individual mutations that mostly don't take off, but some of them did take off. And in particular, you can see in blue, the 614G. So this is quite well known, but other ones popped up as well. Firstly, this 614G in association with a G485R mutation. So this arose within a meat processing abattoir, quite a few cases occurring. Then another mutation here, S477N, which was quite small numbers initially, and arriving, if you can see, these from overseas travellers returning. So this was arriving into the country, unlike the 485R. So this abattoir outbreak sort of arose spontaneously within the abattoir and then spread into the community. This 477N occurred in conjunction with the 614G, and it sort of bubbled along at first, but as we've found out, this was the strain that's emerged, broke out of quarantine into the community, and these are all community transmissions. And now this is the dominant virus. So 98% of all sequences in Australia at the moment have this particular mutation. A few other ones were found. The N501Y. This mutation also arrived from a traveller and then established as a cluster as it broke out from quarantine. And this also occurs in conjunction with the 614G. We still see some 614G only strains, but pretty much now dominantly across the country it's 477N. So just to put some more simple summary on that, there's a couple of mutations that are really dominating. And if we compare across the whole of the world's spike sequences that are accumulating in GISAID and try and understand, the penetration of these mutations, it seems that in Victoria, we have the dominant number of these spike mutations that have arisen. So pretty much all of them are in conjunction with D614G. There's more than 3,000 and now nearly more than 4,000 documented S477N across Australia. G485R, 40 of these, and the N501Y, 34. So I think these were particularly of interest to us for a couple of reasons. Occur in the receptor binding motif of the receptor binding domain. And they had clear capability of expanding and transmitting. So they seem to be selected for efficient transmission. So I'm going to talk to you firstly about this one here, N501Y. So this one was of interest to us because when we looked at the sequences, there was, at the time that we first observed this, only one other that was identified in a patient from the US. It had some different mutations elsewhere in the genome apart from the N501Y. So the Victorian isolates, one here that's got slightly different, it's a synonymous mutation anyway, so it lacks that particular nucleotide change, but otherwise all of these are the same virus and different to the one that is in the database. But curiously, similar position to one other change in this N501T, which is the virus that was isolated infecting mink in the Netherlands. So the other thing that was quite curious to us about this particular strain is that it was the same location of mutation in the spike as this SARS-CoV-2 adapted, mouse-adapted virus infecting BALB/c mice from a group in China. So a science paper out at the end of July where they took isolate and then did 6 rounds of forced passage through mice, and they acquired a strain of virus that had just 5 mutations, but one of them here was this 501 N to Y mutation after 6 passages. So that mutation, if we look at that, it increases the affinity with interaction with the human ACE2, but importantly, it also increases the affinity of binding with mouse ACE2. So we wondered whether this particular combination of mutations might in fact drive a potential capacity of this isolate to infect other species, in particular mice, because we didn't have a mouse model. So doing molecular docking studies, so Lee Hardy in my group learned how to do all these docking work with his the new structures coming out with full-length spike and full-length ACE sequence. And if we look at the delta G, so a lower delta G predicts higher affinity here, better affinity. So human with the wild type spike, the Wuhan index strain. And as we go up across the different species, by the time we get to mouse, there's very, very low affinity here for mouse compared with the human. So when we look at the double mutation, so the delta G, so anything below the line here is going to increase the interaction of the increased interaction with the ACE2 of the particular species. So this mutation predicted to increase the affinity with human ACE2 and quite a few other species as well. The goat and sheep, particularly the horse, donkey, but the mouse was also predicted, and rat, to have significantly higher affinity. So we tested this using the 2 isolates. So we isolated this virus from patients, so the N501Y double mutation with the 614G, and compared this with the index strain. So the first isolate prepared VIC01. And we're doing a competition with the human soluble ACE2 and a mouse soluble ACE2, looking at the capacity of these to reduce the plaques here. So human ACE could inhibit the infection at high concentrations with the index strain, but also the mutant strain. But mouse ACE was unable to inhibit the infection and formation of plaques, even at highest concentration. However, with the mouse, or this VIC2089 strain, so the N501Y mutation, this was able to inhibit the binding of ACE2. So we passed this virus to our collaborators across the street at the Walter and Eliza Hall Institute, Mark Pellegrini, James Cooney, and Utilising, and they infected mice, both Belb C mice and C57 Black 6 mice, with this N501Y strain, and as a clinical isolate, so no adaptation, this was able to immediately infect and produce infectious virus at quite high titer, so 10 to the 6 in the very first experiment, 3 days after infection, whereas the normal wild type mouse with an N at the position of 501 in each of these strains was unable to infect. So this is a clinical isolate able to infect into mice straight away. So that's just comparing the spike mutations that we have in this clinical isolate against other isolates. So the initial strain from the Chinese group, where they engineered or adapted the virus, they had the 501Y. They had another change at the this A653V. So something also within the same vicinity, but the Barrick group have produced 2 other mouse-adapted strains. So these have got a series of mutations at very adjacent positions, 499 and 498. So these are also quite effective at allowing infection within the mouse, but they've done further adaptations, and another particular change at 493, a Q to K to lysine has occurred that's assisted in the ability of this virus to infect after 10 passages and cause pathogenesis. So I should say that we don't notice pathogenesis with our particular isolate. And there's another isolate that's also got a change in similar positions within spike. So looking at the impact of that particular mutation on receptor binding domain interaction by interferometry, we see that it's able to bind with higher affinity than the index strain. And more importantly, it has a very slow off rate, much slower dissociation in this analysis. So maybe this increased dwell time is, so the binding of virus at slightly higher affinity, it's not as high as with human, but is actually perhaps associated for a longer period of time because of this particular tyrosine interactions that enable this to facilitate infection. If we look at the impact of that in terms of replication kinetics, comparing these 2 strains, Viro cells comparing the index and the mutant strain, essentially the same. And similarly with HeLa cells transduced with high expression of ACE2, human ACE2 receptor, we see that there's pretty much equal replication kinetics. So the virus doesn't seem to alter too much else about the replication of the virus with that single change in the spike. But as we look across other parts of the virus that have evolved and changed during this process of forced adaptation, we see that the Victorian virus here, so green indicates a particular change. It has some other changes in the 5' untranslated region, NSP3, NSP12, in the matrix, ORF8, and a few other changes down in the nucleoprotein. So these are all different positions and locations to the mouse-adapted strains. So we're not sure whether these other strains that induce more evident immunopathology, these other changes might in fact have some bearing on that. So the Barrick strain that was further adapted through 10 passages acquired additional mutations. Some of these, in the non-structural proteins, but not the same position as any of ours. And this is another isolate that's published with changes. So what about these other mutations here? What's the impact of those? Are they also predicted to have perhaps an expanded tropism and the potential to potentially spill back into other species? So, Linfar Wang has presented in the GVN forum, giving the potential, or raising the potential of viruses to not only out of the environment from bat to pangolin or whatever, then into human, but the possibility of spilling back into an environmental host. So just refreshing again, showing you that this 501Y was successful in infecting mouse with this increase here, predicted increase in affinity by a small amount. But you can see that there's many other species here that are also within the same kind of affinity range, and there's a potential for these to also maybe infect with this particular strain, but untested at this time. The other mutations, so 477N, when we do the docking studies with this, it doesn't predict a particularly high affinity with mouse, or a little bit higher with rat, but some of the other species, particularly horse, cat, dog, are also potentially increased in their affinity, but that doesn't necessarily mean anything. It's only an in silico sort of modelling study. And then the other one, the 485R, similarly, several of these predict an increased affinity, again, not so much with the mouse. So just to put this to the test, particularly interesting for us was whether this 477N, double mutant with D614G, could in fact transfer and infect in mice. So again, the Pellegrini group at Walter Institute taking these isolates. We have isolates now from each of these mutant strains. It's only the N501Y that is infectious within the mice, and not the 477N, not the 485R isolated from the abattoir population initially. So in terms of infection of mice, there's only one of those strains so far that has been useful, but that turns out to be a very, very useful thing because we didn't have a small animal model, but now we have a natural isolate isolated straight out of patients that is capable of infecting mice, and we're using this, as I'll show you later, to test vaccine candidates, and also could be quite useful testing antiviral drug compounds. So what's the the relevance of this for antibody-mediated immunity against these viruses? So one thing to say is that there is a slow, ongoing mutation of the virus without any immune pressure. So these are presumably selected only for transmission efficiency. We don't fully understand what drives the selection of some of these. It could only be simply a founder effect. But when we look at the potential impact for these mutations in the receptor binding domain to alter the sensitivity to convalescent plasma taken from early patients, so these are all going to be index strain infections, testing against the 501Y mutation, we see that this convalescent plasma that is quite effective at neutralizing the index strain. When coming and testing against the mutant strain, there's much more resistance of this strain for neutralization with antibody from convalescent plasma from the index strain. And similarly, index strain convalescent plasma against the 477N has a couple of logs difference, but is perhaps more capable of neutralizing this particular strain, which is the dominant strain in Australia. So there is some impact, but we're not quite sure exactly the significance of this for the potential for convalescent plasma to be protective for the subsequent encounter with these mutant viruses. Now, if we look using pseudovirus reporter assays, sort of been working hard to generate reagents to test in vaccine studies. So vaccine studies underway with candidates in Australia, stabilised spike trimer vaccine, and also in-house we've got virus-like particle vaccines and receptor binding domain vaccines of various kinds. So testing against this particular strain, the 501-614 double mutant seems to be a very important component of understanding the potency of vaccines for protection against something that is a virus that has circulated within our community right here in Melbourne. So firstly, let's antibodies by Collaborators at Purcell. When we compare the neutralization, the IC50 values, some of these monoclonal antibodies that neutralize at very potent IC50 values, reasonably potent anyway, they're way less potent against the 501Y. So as a monoclonal antibody, some of this particular mutant, 501.614, is quite a difficult virus to neutralize in this pseudovirus reporter neutralization assay. So looking more widely at convalescent plasma, so convalescent plasma now testing against the, in a microneutralization assay, against the 477N strain. So this is the dominant strain in Australia, but we're sort of quite pleased or happy to see that the neutralization of convalescent plasma is a bit more capable against this particular mutant strain. So it's more potent against the wild type index Wuhan spike, about one or two dilutions less capable against the mutant for high neutralizing sera. So some of these are not really great. So actually these are samples that have passed the test to go on for convalescent plasma therapy. But in this assay, it's not looking like they're that hot. This is a surrogate virus neutralization assay with the same plasma, just to give some reference. This is the Linfar-Wang assay, we call it here. So bit of a further deeper look with that particular mutant, the dominant mutant. It looks like it's a bit more accessible with virus, and the monoclonal antibodies with the this pseudovirus 477.614. Similarly, we see many of these monoclonals have really reduced their activity against this particular mutant strain. So what about vaccines? So one of the vaccine trials underway at the Doherty at the moment. It's a candidate vaccine, so I don't want to go into too much detail about what it is, but some of the early results have been quite pleasing, testing using microneutralization against these three quite important and dominating mutations in our community. And pleasingly, vaccination seems to achieve pretty equal level of neutralization in a microneutralization assay against these different viruses. So the convalescent plasma is a bit more limited in its breadth of activity. Monoclonal antibodies, obviously extremely limited, some of them ineffective, but vaccine-induced antibodies with these vaccines that we're making to try and address this particular issue to develop breadth seem to catch these three important strains in our community so far. So in summary, the investments in pandemic preparedness have been really critical in our response to COVID-19, just to monitor and trace the virus, follow it, look at its genomic sequences, and in particular, linking diagnostics with full genome sequencing has led to us recognizing important mutations in the spike of the virus at the receptor binding domain, in the receptor binding motif, that impact on affinity and tropism of virus, and leading to a mouse infectious clinical isolate, which is potentially a concern for spillback into other species, but in a research setting is actually extremely useful to have this natural isolate that we, it's not a gain of function mutation or anything that we have to explain and justify to the ethics committees, but something that is a real concern as a circulating isolate, and a useful isolate for testing vaccines in challenge studies, something that's underway this weekend, and also testing potential therapeutics and antibody therapies particularly. So these spike mutants are emerging. It seems like they're emerging at a very high rate. We're not sure whether we're doing something different, or it's just the very deep sequencing that we're conducting, or the selection potentially of virus strains that are transmitting very efficiently because of the need to break out from this strict social distancing that has been applied. But these spike mutants are emerging, and there are important implications for vaccines, therapeutic neutralizing monoclonal and polyclonal antibodies, and also potentially any COVID-2 antivirals targeting the spike protein. But importantly, going back to the original slides where I showed you this important 501 mouse infectious virus strain was completely extinguished by strict public health measures. So social distancing, mask wearing, and all these things have taken care of these strains and snuffing them out. So including the 477N strain that's dominated the landscape in Australia for genomics has pretty much been put to the sword by these simple public health measures. But nonetheless, neutralizing antibody breadth is an important issue. We see that these mutations do arise. They do have an impact on antibody protection. And we're monitoring this and following this in Australian vaccine clinical trials that are underway as we speak now. So I'd like to really thank my vast array of collaborators, mostly in the Doherty Institute. So all of these people have been very important in each and every step. So Sharon Lewin, the director, Mike Hatton, Julian Druce doing the diagnostics, Julian making the isolates, the clinical isolates, all the sequencing done by Ben Howden and his team over here from the genomics. A huge effort to sequence all of these full-length genomes and deposit all of those sequences into a publicly available databases so that you can follow and find these sequences yourself, like GISAID. My lab staff that have conducted all these cloning experiments to capture and produce soluble spike proteins and pseudoviruses and conduct these assays. Cantor and her team, others, actually Amy Chung, is she hopefully on there? I might have left Amy off, but clinicians, Terry Nolan driving the vaccine clinical trials, Weihai, Mark Pellegrini and his team doing the mouse infections, soluble ACE and convalescent plasma coming from Monash University collaborators, and some structural studies from Michael Parker and people at Bio21. I'll end it there. Let me see if I can see people.
Yeah, thank you so much, Damian. This was really this has been a great talk, and which really illustrates the power, I would say, of molecular epidemiology and real combining different strategies. Very impressive, and also, as you pointed out, some worrying news. But so we have the first question. Can you see the question, Damian?
Not yet.
Q&A.
Okay, there it is. Right, okay. So David, does 614G change have any functional relevance or interactions with, yes. So this is a great question. I didn't have time to go into this. So using the pseudoviruses, we've been able to analyze these mutations on their own and in conjunction with the 614G. So the 501Y in particular benefits a lot in its affinity with 614G. So the combination, when we do neutralization studies and infections, those two mutations seem to work together, and it's important to have both of them to have the full activity of that particular isolate. So, I think it was interesting that I showed it the mouse-adapted virus that emerged in the experiment in China had that 501Y mutation, but also another change in the same vicinity of the spike protein as the 614G. It's a different amino acid, but maybe it also has a similar effect in changing the presentation and access of the receptor binding domain itself.
Thank you very much. Maybe to follow up on this question, I mean, this is an obvious point, but from your work and also from the other ongoing studies, seems that we keep on a positioning where there is evidence for these mutations strongly modifying the tropism and enhancing the capacity of infection, but there is no evidence for increased or decreased virulence of these strains. Would you confirm this?
So at this point, yes, there's no evidence of disease in the mice. So it's early days for that part of the study. And in terms of understanding the clinical course, so this is one thing that's still, if you like, a deficiency in our approach. We don't have the ability to connect across to the patient. So the ethics and confidentiality don't really allow us to know too much about the role of these mutations, particularly those that are in relatively smaller numbers, the 60s or 80s. There is a suspicion that the S477N might have a longer period of time of asymptomatic infection, which might make sense, but we haven't yet been able to actually connect the clinical data to those particular mutations. But it's not a higher death rate or anything like that. That's
but your point is very important. We absolutely need these correlations in the future, and also joining effort on this mutation to collect data from different parts of the world, because this is obviously a key point. And but regarding as you pointed out, I mean, these mouse infecting isolates are extremely interesting for research. Very much worrying regarding the potential for dissemination of the virus. I have a naive question. I apologize for this, but these findings, such findings, I would say, have been made for other coronaviruses, I mean, including obviously SARS-CoV-1, but other, or is it something which in your view is specific for SARS-CoV-2?
No, mouse-adapted strains have occurred, in other species. So, I think that, in the SARS-CoV-1 and less so with MERS, but, my awareness of that is not as high. I'm sort of very aware of the SARS story. But the possibility of establishing new reservoirs is very real. I think that Linfar's point that he made in the GVN webinar about the potential for us now, high frequency of infection to actually seed infection into the domestic animal species, or even species that might be turning up in abattoirs, for example, and passing the virus back. So cows or goats seem to be quite predicted to be quite susceptible to the infection. So if that can happen, it's a real great concern, actually eliminating the virus out of the community. So
absolutely. You have a question?
What about the impact of these mutations on new animal reservoirs for SARS-CoV-2? Yeah, exactly what we're talking about. So I think this is something that's unknown. And so what we do recognise with the clinical isolate passed straight into the mouse, that the mouse is actually able to clear that virus after about 9 days. It's it deals with the infection. There's no great inflammatory disease, not in the C57 Black 6 strain anyway, but we're mostly looking at relatively younger mice. This phenomenon might occur in older animals. We haven't had a chance to test that, but it seems that there is potential to at least transiently infect animal species, at least a mouse, with potentially these clinical isolates. So it's been established with cats, and I think dogs, and whether those species, will sustain a longer term transmission chains is really a question that I think needs to be thought about and addressed.
And do you, to follow up on this, because this is a very important point, again, are you aware of ongoing studies on this specific topic worldwide? I mean, in the One Health, I would say, context?
So I'm not familiar with anyone actually doing the work. You know, we've kicked it around whether it's worth doing an investigation where we transfect ACE2 from different species into cells and see whether, you know, it's feasible. But these potentially gain of function experiments need to be thought about pretty carefully before you know, you really embark on those things. So I know in Australia we have very strict regulations transferring viruses into domestic and animal species.
No, but that was, Damian, if I may, I mean, because gain of function is a very sensitive topic, as you said, but just by looking within the infected, the present reservoir, I mean, you find this combination of mutation, for example, you see?
So that's a good question. So I don't know that anyone's actually found that combination of mutations. So the mink in the Netherlands, there was, I think, a talk in the GVN series, somebody covered that, and I wasn't quick enough to see. It was probably on quite a late hour or early hour for me here in Australia, whether that particular, the 614G mutation went along with the 501T mutation. I don't think it did. I think it was just that single change. But, you know, we are seeing occasionally now additional second changes occurring within the receptor binding motif. So the 477N, as we're seeing little runs of other amino acids that also change. So, you know, 2 changes in the receptor binding domain in patients. You know, these could have some significance in terms of potential for spill back into other animals. So, you know, it's sort of pretty easy to see how this kind of virus in populations of species that have, you know, high density, like the bats and other animals within, you know, other scenarios, could actually spawn new strains. And I think that, you know, Linfar's theory about these things having capacity to jump species pretty readily, you know, really does look, in our case, to hold some water. You know, there's many more mutations in that significant region than I expected to find in our community.
Well, thank you very much. I believe that we will end up this seminar now. Well, thank you so much, Damian. It has been great. Thank you very much.
Thank you, Christian, and whoever has managed to stay up so get up or stay up so late here in Australia. It's going on midnight, nearly Friday here. So
okay, and bye to everybody. Thank you.
Thank you.
Thank you. Bye.